Welcome back to Microscope. Today we are going to know about tools and techniques. So we all know what is a microscope. We can see a minute images which can't be visualized by an eyes. That images can be seen through an microscope. So in this microscope has been become so much advanced that everyone can see highly magnified image structure and very minute structure can be observed, but also we can visualize the three-dimensional structure of such objects by a powerful tool that is a microscope. And how can we visualize? First we will prepare any slide of a plant tissue of any microorganism like virus, fungi, etc. And then we will just place that slide above the stage, and by the object we can observe by keeping our eye on the eyepiece. So there we can see and observe and magnify the images as we want according to our eye adjustment, and we can visualize the things very easily. So using powerful electron microscope technique, even the DNA molecule or bacteria virus that have been visualized. So first microscope, do you know who in date backs in 1665 and who designed the first microscope? So the Robert Hooke, he designed a simple microscope using a combination of magnifying glasses and observed the slice of cork and coined the term cellula or cell to the honeycomb like structure. Matthias and Jacoba Sheldon and Theodore Schwann proposed cell theory in 1838 on the basis of observation of the cell in plants and animals. So you know what is magnification and resolution. So let's understand the principle on which the techniques of the microscope is based on. The two optical properties are extremely important and for optical instruments like microscope. So one is the power of the magnifying and the other is the ability to resolve it. So magnification or magnifying power of microscope is the ability by which the retinal image size that can be increased. In simple terms, we can say that the size of the retina image, with the help of the microscope, we can see upon the size of the retinal image without using microscope. So you must have studied in physics that a magnification of a lens is measured as per the following formula, like m is equal to f upon f minus d, where f is the focal length of the lens and d is the distance of the object from the lens. That is focal length of the lens upon focal length of the lens minus the distance of an object from the lens. So this microscope which is used in laboratory, it is a compound microscope in which the two set of the lens are there, and one is the objective lens and which remain closed and the object is seen, and the other is the eyepiece through which the observer, that means we can see by the eyepiece. This is needless to mention that the objective lens or eyepiece and the eye of the observer have been in the same line for passage of the light to see the magnified images of the object. In simple word, magnification of the microscope is a product of magnifying the power of the objective lens. So magnification power of eyepiece, that is m0 into me. That is the formula here. So resolving power is another important property of microscope. That is the ability to form a separate image of two objects situated very close to each other. It can be measured by smallest distance between 2 points. What is functioning of light microscope? Do you know what is a light microscope? A compound microscope consists of a base of which a stage is filtered with central hole attached to the base in the arm to which the body tube is fitted in such a way that it aligns with the hole of the stage. At the lower end of the body tube, a nose piece is fitted on which the two or the four objective lens may be present. By rotating the nose piece, one of the objective lens can be placed above the hole present on the stage where the object to be seen is placed on a glass slide. At the upper end of the body tube, an eyepiece is fitted through which an observer, that means you can see the under the microscope, and these adjustments screwed, that is on the arm, which facilitate in adjusting the distance of the objective lens from the objective present on the stage. So below the stage there is a source of light, which means a reflective mirror or a bulb to illuminate the object and facilitate the formation of image through the objective lens and eyepiece. In addition to this, there is a condenser that is present between the light source and the stages, which is important for focusing light on the object. You might have observed that both objective lens and eyepiece are of different magnifying power. In a microscope, the eyepiece has a magnifying power of 10x or 15x, and different objective lens fitted on the nose piece or eyepiece is 4x, 10x or 42, 45x or 100x. So this technique of microscope just discussed is called a bright field microscope. As the light is used as illuminate the object to be seen, and therefore in order to distinguish different region of the object, the same is stained with specific dye or stain. Caramine, eosine, safranine, methylene blue, gymsust, etc. They are the few such stain that is commonly used for the light microscope. There is an eye magnifying image, and there is an eyepiece. The projector lens are present, and objectives and specimen, and then condenser light and the light source, that is the below. At the very below of the microscope there is a light source. Then a different form of microscope. If we study the minute details of internal organization of the tissue cell, and it is so diverse that it cannot be achieved by light microscope only. Therefore, by making one of the other kind of quite diverse form of microscope are used, and in one such, light falling on the object from the central condenser is blocked by distance, and illumination of the object is done by an oblique light beam that is reflected from the slide, and the image is illuminated against the dark background. Such microscope is known as the dark field microscope. There is a mitochondria, nuclei, vacuole, etc. that can be easily detected using this. So in different form called the phase contrast microscope, the wave amplitude and the phase of light that passes through the transparent object is changed. This change depends on the density of the part of the object or the specimen. So such a change is more in the area when density is comparatively high, and as a result of which very construct of different region of the object can be seen. It is especially helpful in the study of cell organization and chromosome. Staining of object or specimen with some specific dye is routine done. There are some specialized type of dyes. Example, acrylyl orange, bisbenzamide, myrocyanin, etc. They are the fluorophores. These dyes are capable of emitting light or long wavelength after being illuminated, a property called fluorescence. So as a result, the fluorophore stained object look more illuminated and of different color depending on the dye using a fluorescence microscope. The same principle is applied. So object to be seen is stained with fluorophore to study a specific part of the organelle or molecule. After illuminating the object under fluorescent microscope, the specifically stained region is easily seen or observed. So this is helpful in identifying the bacteria or virus to know the cause and infection of immunodiagnosis. Then electron microscope. It is highly sophisticated technique in which the object is to be studied and bombarded with electron beam with an approximately 100,000 times shorter in wavelength than visible light. The electron beam in electron microscope, it magnifies the image with the help of the electromagnetic lens. The entire passage of the electron is in vacuum, and generated image is viewed on the fluorescent screen and not through the eyepiece, owing to a very short wavelength of the electron. The image produced by electron microscope is of very high resolution. Two types of electron microscope are used: transmission electron microscope and scanning electron microscope. So the transmission electron microscope, it is an ultra thin heavy metal salt of lead and tungsten. It is coated section of the object of specimen is placed in such a way that the electron beam passes through it to create an image. So in other technique of electron microscope, it reflects the electron beam from the gold or platinum coated surface of the object, creating the image. So in this technique, highly magnified or resolving image of the object surface is generated. Therefore, it is known as a scanning electron microscope. The confocal microscope, it is useful in resolving the detailed structure within fixed cell tissue and giving shape image of the object. To examine an object using confocal microscope, it is first fluorescently labeled and then analyzed under a confocal microscope in high resolution. So this was all about the microscope. Now moving on to centrifugation. What is centrifugation? You have studied about various biomolecules like protein, nucleic acid, etc. present in the cell and all living organism. So to study this biomolecule, you need to isolate them by using one or the other separation technique. I have made a short on the centrifuge. You can go and visit and watch the video of short on the centrifuge. Thus, what are the steps for centrifugation? And centrifugation is one such technique in which the particle or the molecule, they are separated based on the density under the influence of gravitational force. We spin them in a solution around an axis at a high speed using a centrifugal force. The equipment used is called centrifuge, and which is of different type depending on its use. So it consists of a base, a rotating container that's spinning the vessel or rotator, and a lead. So the spinning vessel continue serve centrifuge tube and the cell is extracted or mixture is taken in the centrifuge tube and allowing to spin at a desired speed, revolution per minute, that is RPM, for a specific period of time, leading to the settlement of a particle material at the bottom of the centrifuge tube. What are the various type of centrifuge? Based on the principle and application, there are differential centrifugation, then density gradient centrifugation, and ultracentrifugation. So differential centrifugation, it is based upon the difference in the sedimentation rate, like centrifugal force or a particle of different size and density. It is used to separate large cellular structure and nuclear fraction, mitochondria, chloroplast, and large protein. Then density gradient centrifugation, in order to separate the biological particle of similar size but differing in densities, one can use density gradient centrifugation. In this type of centrifugation, density gradient is developed in the centrifugal tube. Depending upon the density, different molecule get sedimented at different level. So the heavier molecule move outward and the lighter one remain in the inner part of the centrifugal tube. So the greater the difference in density, the faster will they move. Then ultracentrifugation, when the centrifugation is carried out at very high speed, like 100,000 x per gram per g or more to separate the molecule, it is known as the ultracentrifugation. This is commonly used in tabletop clinical centrifugation or microfuge or high speed centrifugation or ultracentrifugation. The next one is electrophoresis. Now it is a method of separation of on the basis of the charge to mass ratio of a macromolecule under the influence of electric field. So electrophoresis is means to bear electrons. So it is a Greek word. So the prefix like electro, that means electricity, and it required to migrate a molecule and means migration or movement. The suffix phoresis means migration or movement. So this was observed in 1807 by Russian professor Peter Stekhoff and Frederick Ries. They noticed the migration of the clay particle that disperse in the water in presence of a constant electric field. So what is the principle of this electrophoresis? Electrophoresis, many important biological molecules such as the nucleotide, DNA, RNA, peptide, and protein, they bear ionizable group and therefore at any given pH exists in the solution an electrically charged species either as cation or anion. Under this influence of an electric field, these particles will migrate either toward the cathode or anode depending on their net charge. The mobility of the molecule is inversely proportional in its size and directly proportional to its charge, allowing them to be separated from one another. Now, what is an agarose gel electrophoresis? There is a gel in the matrix of agarose molecule that held together by hydrogen bond and form a tiny pore. So the gel form a DNA separation. They are often made out of polysaccharide. They are called as agarose, and which commonly is a dry or powdered flex. And when the agarose is heated in buffer and allowed to cool, it will form a solid and slightly squishy liquid, a gel. So this gel is tab of jelly like material that is placed in a gel box, and one end of the box is connected to a positive electrode and another end is with a negative electrode. So the gel box is filled with a salt containing buffer solution that can conduct current. So the end of the gel with the well is positioned toward the negative electrode. The other end of the gel is positioned toward the positive electrode to which the DNA fragment will migrate. Then DNA molecules are negatively charged. The gel electrophoresis of DNA fragments separate them on the basis of the size only. So using the electrophoresis, we can check the different DNA fragment present in the sample and determine their absolute size with the help of the DNA ladder, which is made up of DNA fragment of the known size. So when the power is turned on, the current began to flow through the gel, and DNA molecule have a negative charge due to the presence of the phosphate group in their sugar phosphate backbone. Therefore, they move toward the matrix of the gel toward the positive electrode, that is anode. So the voltage of running of the agarose DNA gel lies in the range of 80 to 120 volt. As the electric current is applied, shorter pieces of DNA travel through the pores of the gel matrix and faster than longer one. So thus the longer pieces of DNA remain near the well, while the shorter pieces of DNA they are close to the positive end of the gel. Then visualization of the DNA fragment. The equipment used for visualization of the target DNA is UV transilluminator. Ethidium bromide is most commonly used for staining the DNA. This stain can be mixed in the gel mixture in electrophoresis buffer, or the gel is stained after it is run. The molecule of the ethidium bromide intercalates within the DNA based on the fluorescence under UV light. Despite its advantages, ethidium bromide is a potentially carcinogen, so it must be handled with great care. Then PAGE, that is polyacrylamide gel electrophoresis. The PAGE is an analytical method that is used to separate the compound of protein mixture based on the size and to provide a uniform charge of the protein molecule. An anion detergent called the sodium dodecyl sulfate, that is SDS, it is used to bind the protein and give them a negatively charge. Protein are then separated electrophoretically according to their size using a gel matrix that made a polyacrylamide in an electric field. So polyacrylamide is produced as a result of the polymerization reaction between acrylamide and N,N-methylene bisacrylamide, that is used as a catalyst. So degree of polymerization or cross-linked can be controlled by adjusting the concentration of acrylamide and bis. So more the crossing link, the harder the gel. Harder macromolecule when they travel through the gel, through the PAGE, so thus affecting the resolution of the separation. So the gel, that is 4 to 8% acrylamide, allow high molecular weight molecule to migrate faster than the gel, while harder gel, that is 12 to 20% acrylamide, restricting the migration or a large molecule and selectively allow small one to move through the gel. There is a tracking dye. Do you know what is a tracking dye? As DNA and RNA protein, they are made up of mostly colorless. These progress through the gel through electrophoresis that cannot be easily followed. So anion dye, they are known as electrophoretic mobility, are therefore usually included in the sample buffer. A very common tracking dye is bromophenol blue. This dye is colored at alkaline and neutral pH and is a small negatively charged molecule that moves towards the anode, and it is colored at alkali and neutral pH. And being a highly mobile molecule, it moves ahead of most of the protein and nucleic acid. As it reaches to the anodic end of the electrophoresis medium, electrophoresis is stopped. Another common tracking dye They are xylene and canol, which has lower the mobility, and orange G, which has a high mobility. Then the visualization of protein on the gel. COMSO or brilliant blue R250 is the most popular protein state, and it is anion dye with non-specific binding to the protein. So protein in the gel are fixed by methanol and acetic acid, and simultaneously they are stained. The excess dye is incorporated into the gel and can be removed by destaining with the same solution without the dye. So the protein are detected as blue band on a clear background. After the visualization by the staining, the protein specific technique, the size of the protein can be calculated by comparing its migration distance with that of the known molecular weight ladder. So for knowing such concepts, follow Biomindscape. Thanks for watching. Do like, share and subscribe.